Tonight's event is presented by Royal Botanic Gardens Victoria as part of Survive and Thrive, the program for National Science Week. We're supported by the Inspiring Victoria program, and this year's Science Week theme is Species Survival. I'm Alison Vaughan, the lucky person who gets to be the collections manager of the wonderful herbarium collection here. And it's my very great pleasure to talk to you about the herbarium collection for probably close to the next hour. Before I begin my talk, I would like to acknowledge the traditional owners of the land we're on tonight. We're on Wurundjeri Woi-wurrung country. I pay my respects to their elders past and present, and to any Aboriginal and Torres Strait Islander people here tonight. In talking about the herbarium and the science that we do here, I also want to acknowledge that we're talking very specifically about the Western botanical system of knowledge, and I'd like to recognise that the many peoples of this country have had rich, deep and long histories of knowing, understanding and relating to plants, algae and fungi, and those knowledge systems sit alongside the knowledge system that we use here in the herbarium. So this evening I'm going to talk about the herbarium collection here, which underpins the garden's science and conservation work. The first part of my talk will be about the role the herbarium plays in supporting that science and conservation work that does support species survival, and probably with a bit of a local focus. I'll then talk to you a bit about the magnificent global collection that we have here in the herbarium. But I also, as well as talking about the specimens, I do also want to talk about some of the people behind it, the people who made this collection come together and make it the amazing resource that it is. But I will start at the beginning and talk about what a herbarium is. So unlike our cousins' museums, we don't sit very high in the public imagination. A lot of people have no idea what a herbarium is and haven't heard one, let alone been inside one. So they are a bit of a mystery, but even though they're not, widely known, they are very widely used. And every time that you use a species' botanical name, a plant's botanical name, you are referencing the work of herbaria. This is where botanical names of plants are born. I'll talk a little bit more about that later. But at the most fundamental level, a herbarium is a collection of preserved plants, algae and fungi. The majority of specimens in herbaria are pressed seed plants that are preserved flat, mounted onto card that's about A3 in size. There's some examples over on the side here that you can have a look at later. But we do also have specimens preserved in ethanol, woody fruits and cones that are stored in boxes, and lichens, mosses and fungi that are stored in packets to preserve their three-dimensional shape. So around the world, there are more than 3,300 active herbaria, all countries. And between them, they house an estimated 390 million examples of life on Earth. The National Herbarium here in Victoria was founded in 1853 by Ferdinand Müller. Müller was a former director of the gardens and he was the first government botanist of the state. Our herbarium here is the oldest scientific institution in Victoria, and it's the first herbarium in Australia. The original herbarium building here was, and sorry about the greenish tinge you've got there, was over near the Shrine of Remembrance, sort of a bit beyond where that toilet block over the road is now. But because herbaria continually grow, the collections are always getting bigger. They do outgrow their homes. So this one had the extension built that you can see, but then in 1934, it moved into the building that we're in now. So this building, the Art Deco part of it, was a gift to the state, a centenary gift to Victoria by Sir Macpherson Robertson, who you might know of. He was the confectioner of Cherry Ripe and Freddo Frog fame. He has McRob's Bridge and the girls' school named after him as well. The herbarium collection kept growing, and then in 1989 it moved into this circular extension where it remains today. So this extension is just on the back of the building we're in at the moment. It's very much outgrown this building too. We have boxes and trolleys of boxes everywhere. But this herbarium here is home to the State Botanical Collection, which comprises extensive archives and botanical library, a large collection of original botanical artwork, and the 1.5 million preserved plant, algae and fungi specimens. Although it's less well known than the beautiful gardens that it sits within, the herbarium is very central to the work of Royal Botanic Gardens Victoria. Plants, algae and fungi are fundamental to life on Earth. We can't survive without them, and if we want to ensure the survival of not only our species but the myriad species we share this planet with, then we urgently need to understand those plants, algae and fungi on which we rely. So the herbarium collection here in Melbourne and herbaria more generally around the world help us do that. Each one of those 1.5 million specimens in the herbarium is a permanent record of the presence of an organism at a particular place and time. They're like botanical time capsules. Together, all of those specimens make up a really powerful and big picture of biodiversity, both past and present. And as the collection grows, that picture of biodiversity becomes clearer. Every new specimen coming into the collection improves our understanding of a species, its biology, its ecology, and it gives us more information to protect that species into the future. Like any collection, whether it's a museum, a library, archives, an art gallery, we care for it to prolong its useful life, and this is very much a collection that is used on a daily basis. We do that, we care for that collection in a number of different ways. So one of the first steps is to mount the specimens. So that is to attach them to archival card, and that gives them the support that they need to be examined by botanists without being damaged. Now, over this display case closest to me over on the wall has got some great examples of specimens mounted by our wonderful herbarium volunteers just in this past week. So we have about 54 volunteers at the moment who each dedicate some time each week to come in and do this really painstaking but incredibly important work. So you can have a look at that, and we've got at least one of our wonderful volunteers in the audience tonight. We host visiting researchers who use the collection. We database the specimens so that the collecting information is accessible for research. We send specimens and samples of them, tissue samples, on loan to researchers at other herbaria around the world for them to study. And when resources permit, we photograph the specimens to provide a digital proxy that can facilitate research, just open up that collection a bit more. But perhaps the most fundamental way in which we continue to enhance the collection is by keeping it up to date. And we do that through continued collecting. So in order to flesh out that picture of biodiversity through time, we need to avoid having gaps in the collection, either in terms of space, so geography, through time, or through species. And if we can avoid having those gaps in the collection, then we get this beautiful time series that allows us to study how the physiology of plants changes through time and how their distributions might have changed through time. And then that allows us to predict how they might change into the future. But before we can actually undertake any of that work, we need to know what we're talking about. So we need a shared language or agreed names for the plants, algae and fungi that sustain life. At the most fundamental level, herbarium specimens support the science of taxonomy. So that's the describing, naming and classification of species. Herbarium specimens give us the language, the names to talk about the life around us. And once we have those names, we can create a shared inventory of life. And we can exchange information with others, safe in the knowledge that we're talking about the same thing. Before I move on to the next slide, I'd just like you to note that this specimen up here, which was collected in 2019, only has the name acacia on it. So every time a species is described, its name is permanently attached to a specimen somewhere in a herbarium, somewhere in the world. These are what we call type specimens, and sometimes they're considered the most important specimens in herbaria because of that link to the names. The collection here at the National Herbarium of Victoria is the most type-rich in the Southern Hemisphere. We've got a really high proportion of type specimens, and probably a higher proportion than most collections worldwide. Now, I know for all of you it would be handy if botanical names didn't change. It can be frustrating when they do, but they have to sometimes. As our botanical knowledge evolves, as our understanding of the distinctions and relationships between species becomes clearer, as that understanding improves through time, we need to consult those specimens again. So just as letters, documents and manuscripts are the primary sources of historians, herbarium specimens are the primary sources of taxonomists. So you might have noticed that this specimen up here is the same one from 2 slides back, but now it has the name Acacia uraniae on it. And it was only collected in 2019 and described in 2020 by Neville Walsh. So you can see that ongoing collecting not only improves our knowledge of the plants that we already knew about, it can reveal new ones to us as well. So this is quite a lovely story. It was growing on the property, a conservation property in Far East Gippsland by a couple, the Urans, and they couldn't figure out what species of acacia it was. So they sent a sample into the herbarium. Neville Walsh, our now retired senior conservation botanist, immediately could tell that it wasn't an acacia that had been described. And so a collecting trip went out there, collected those specimens in October 2019, and then worked up a paper that was and published the name in 2020. But in between those specimens being collected and the name being published, those devastating bushfires of 2019-20 went through that area. And what was a population of 80 of these plants became a population of 5. So at the time of collecting those plants, they also collected seed for our conservation seed bank, where we store seeds of Victoria's most rare and threatened plants as insurance against extinction. And so luckily, we do have seed of this, and in the case that the bushfires had wiped out the entire population, we would have at least had something to be able to go back and regenerate that population with. But while we're talking about names and the idea of new species, I do just want to pause and again acknowledge that we are just talking about the Western scientific way of knowing plants. So this plant up here, which we might know as Microceras walteri, and that's a name that was published in 1918, it already had a name. It's been known as Murnong to the peoples of the Kulin Nation for millennia. So we are, again, we try and avoid talking too much about discovery these days, or new species, because they're not new. They're new to one way of knowing, but not new. So I mentioned that type specimens are sometimes considered the most important specimens in herbaria, but they're not actually all that much use by themselves. An individual can never encapsulate all the variation within a species. That would be like picking one of us from this room and deciding that all people were like that one person. So we need a broad coverage of plants and the populations that they come from to capture the variation within a species and the different features that appear at different times of the year, like flowers and fruits and buds, which can be really important for identifying that plant. So these species here, these specimens here are all Eucalyptus largiflorens, or black box, and that those differences in where they were collected, the time of year, and the habitat they were growing in are all captured on the collecting labels, which are in the bottom right-hand corner. And so that bit of information, that collecting information, is an absolutely critical part of the specimen. It's not a scientific specimen without that. So cataloguing all of that collecting information on those labels is a really core part of the work of the collections team who look after the day-to-day care of the collection here. We've catalogued just over 2/3 of the collection. So of those 1.5 million specimens, we're probably up to about 1.1 million that have been catalogued. And the millionth record went into the database just about 2 years ago, almost to the date, actually. This here is our treasured long-term staff member, Rita Makeda, who had the honour of databasing that record. Rita's been with us for over, I think, close to 24 years now. Her knowledge of the collection is just extraordinary. And she single-handedly catalogued about 10% of the records in our database. So it was very fitting that Rita had that honour. Once those specimens have been catalogued and georeferenced, so when we transcribe the information, we transcribe its latitude and longitude if it's been recorded by the collector, and if it hasn't been, then we map it ourselves. And so at a really fundamental level, that digitised specimen information gives us the fundamental species distribution maps, so our understanding of what grows where. So every dot on this map here represents a specimen of Xerochrysum viscosum, the sticky everlasting, and each dot is in a herbarium somewhere. These are all the Australasian herbaria, so those dots aren't all from our collection here, but they're from a collection somewhere in Australia or New Zealand. The great thing about having those specimen-based records is if you have a few outliers like you do have on this map, there's a physical specimen that you can go back to and check the identity. You can see if someone made an error in transcription or whether the identification was wrong. So it's really that verifiability of herbarium collections is a really important feature. So as you saw from that last map, our herbarium data, once it's catalogued, is shared with data from other natural history collections. We share it with the Australasian Virtual Herbarium, which is part of the Atlas of Living Australia, and it also goes globally to unite with natural history collection data from around the world in the Global Biodiversity Information Facility. So within these repositories, that merging of collecting data from all around the world creates these really powerful and freely available, it's all free online, these really powerful data sets that enable large-scale analyses that were previously not possible. So the specimens and their data are used in many different ways, and I'll share just a few of them tonight that are directly related to state conservation efforts. So the science team here at the Gardens have done a lot of bushfire recovery work in recent years, including floral assessments going back to bushfire-affected areas to assess the impact of the fires on the plant populations. The herbarium collection is critical to that because it gives us that fundamental record of what has grown where that allows us to go back and see if it's still there. Herbarium specimens also support our conservation seed bank and spore bank. So every time seeds or spores are collected as insurance against extinction, we've got our seed bank and spore bank are in the basement of the extension where they're stored in either about -25 or -80 degrees. Every time they're collected, the botanists collect a herbarium specimen as well. So that enables us to confirm its identity. And those specimens are critical to the utility of those conservation banks because you can't always tell what a species is by looking at its seed and possibly never by looking at spores. But with the herbarium specimens, you can go back and check. So again, if there's ever any doubt about what the species was that's collected, or perhaps that species has been split into two, you've got a specimen to go back to so you can tell what it is. Without the other way you do it would have to be to grow up the plants from seed, and which would be quite a slow way of doing it. Now, we of course don't study species in isolation, and understanding the relationships between them is really important if we want to, say, look at relatives of medicinal plants to see what other species might have useful properties. or if we want to understand how species with shared history have evolved to thrive in different environments. So we might see what their adaptive potential is. So by extracting DNA from herbarium specimens, researchers can get clues about how different species might respond in changing climates. Our conservation geneticists, and we've got Tara Hopley and Laura Simmons up here, they support our seed bank and spore bank by making sure that we maintain genetic diversity in those conservation collections that support species survival. So we don't want to be only collecting from a narrow sort of genetic cohort of an individual species. We want to get as much genetic variation as we can, so we're maintaining the integrity of different populations. So while the focus of our work is the flora of Victoria, the plants, algae and fungi that occur in Victoria, the scope of our collection and the impact of our work is global. So plants don't respect political boundaries, and we need to understand Victoria's biodiversity in a global context. And to that end, we're really fortunate here in Melbourne to have an extraordinary collection of preserved plants, algae and fungi collected outside Australia. The curation of this global collection has been a major priority for us for the last few years. It is a particularly at-risk part of our collection in terms of the way it's stored. Most of it is still unmounted, so it doesn't have that protection. But it's a really remarkable scientific and cultural and historical treasure that many people are surprised to find here in Melbourne. So more than half the specimens in the global collection, so that's about the global collection as a whole is about 500,000 specimens. More than half of those, probably about 300,000, came to us from the private collection of an eminent German botanist named Otto Wilhelm Sonder. So he was a friend and colleague of Ferdinand Müller's, and throughout his career, he amassed a vast collection from collectors all around the globe. All plant, algae and fungi groups and every continent his collection came from. Müller had to spend almost 2 decades convincing the Port Phillip government at the time to purchase this collection, but it finally came here in 1883. At the time, Müller described it as the richest of all private herbaria in existence, and from what we know of it, we don't think that was, hyperbole. It seems to be objectively true, that claim. Unfortunately, most of the collection has remained relatively untouched for the last century. It has been used, but it does remain mostly in these straw boards, which are kind of charming to look at. There's one over there in that far display case. But in terms of conservation standards, they really fail. The specimens are loose in there, their labels are loose, they risk being separated, and the specimens risk physical damage. So before we can fully realise the scientific potential of the global collection, we do need to mount it, to protect it, and to database it so the specimen data is accessible for research. As we slowly undertake that work, we're getting a better understanding of the riches of that collection. It has specimens collected on the earliest European explorations of Southern America and Southern Africa, including specimens like this one collected by Carl Martius in Brazil on a 10,000 kilometre journey between 1817 and 1820. We have specimens dating back to the 1500s, and this specimen is thought to be from the herbarium of Caspar Bohan. Bohan's herbarium specimens are the oldest surviving ones in the world, so they're from the early to mid-1500s. Those of you with a bit of knowledge of taxonomy and nomenclature will know the name Carl Linnaeus, who published Species Plantarum in 1753, which formalised the binomial system of naming plants, so with a genus and a species. That handwriting on the middle name there, the middle line, is his son's handwriting, Linnaeus Jr.'s handwriting. And the handwriting on the top row there is actually what sometimes gets called a pre-Linnaean name. So it's before that binomial genus-species system was formalised. that there is a really cumbersome, long name of a plant, and it's very rare to see those still on herbarium specimens. The global collection has specimens collected on Livingstone's expedition in Africa, so the Zambezi expedition, and specimens collected by Charles Darwin, Alexander von Humboldt, and other distinguished naturalists. So this one you might be able to read in the bottom corner, was collected in Rio by Charles Darwin. So this collection gives us a rich picture of not only Victorian, but Australian and global biodiversity. And it's through the efforts of thousands of passionate individuals that we have this amazing resource today. And I always feel that people are at the heart of everything, and so I do want to spend some time talking about the people who are responsible for this amazing collection coming together. The richness of the herbarium collection is very much in part, very much due to the efforts of Ferdinand Müller himself. So he was the central figure in 19th century Australian botany, and so all specimens most of the specimens from European exploring expeditions came here, and he also undertook extensive travels and collecting throughout the continent himself. So although he was government botanist of Victoria, he had no thought of confining his fieldwork to just this state. He travelled and collected widely. Those dots there with a few outliers that are probably errors that need cleaning up in the middle of the country were all collected by Müller. There's about 25,000 all up that he collected himself. But to help us understand just how central Müller was to the development of the collection, we need to look beyond just his own collections and look at the ones that he facilitated. So you can see here that there's a couple of spikes. There's a big spike there before Müller's death in 1896. The sharp drop-off after his death is due to a variety of factors, and I won't go into those so much tonight. But I do want to talk about this peak right here. So although he collected widely, Müller couldn't. The continent was too vast for him to get all the specimens he needed to examine to understand the flora of the continent. So in 1876, he published this circular in newspapers around the country. We found about 9 different papers so far that it was published in. He was inviting those who may feel interested to contribute plant specimens to the herbarium. And this led to a huge influx of specimens from people all around the country. And that recruitment of new collectors is very likely a key factor in that spike before his death. One of the people who answered Müller's call was Mary Bate. She began collecting at age 25 around her home in Tilba in southern New South Wales, around Gulagurru, Mount Dromedary. Over the next 5 years, she collected 350 specimens for Müller, and they formed this lovely little record of the flora of Gulagurru, Mount Dromedary, that has since been used by the Friends of Gulagurru to survey the current flora of the mountain. Mary was one of over 225 women who collected for Müller, and so we have this, through Müller's efforts at recruiting and maintaining collectors, we have this really large contribution of women to the history of botany in Australia. I think about 12% of the collectors in Müller's network were women, and studies on, say, Kew Gardens in the UK suggest that only about 4% of their collectors were women at the same time. So those personal collections fostered by Müller are absolutely critical to the development of the herbarium. This map is only of the women in Müller's network, and he maintained those connections by writing letters. So it's estimated that he wrote 100,000 letters in his lifetime. And people sometimes lament that Müller had 3 engagements and never married, but I think he was such a workaholic that it was probably not such a bad thing. So, but I do think it's just that incredible drive to engage people in collecting, and I do think that this project of Müller's could really be considered the first citizen science project in the country. Another key figure in the herbarium history is former herbarium director Jim Willis. So he started as a herbarium assistant in 1939 and was employed as a botanist from 1947. He was assistant government botanist and head of the herbarium from 1961. Willis was described as being unfailingly willing to give botanical information to those who asked and proved an inspiration to many. He made and maintained connections with naturalists throughout the country and encouraged their botanical pursuits. He was another avid letter writer, and we've got, as well as Muller's letters in the library up here, we've got all of Jim Willis's letters as well. One collector whose contributions were nurtured by Jim Willis is Eileen Ramsey, and she's one of my favourite collectors. Eileen lived in the soldier settlement up at Redcliffs in far northwest Victoria, up near Mildura. And in 1949, when she was 62, she started plant collecting. And over a 10-year period, she collected about 1,000 plants. And it formed this, a little bit like Mary Bates' collection, but on a slightly larger scale, a really important record of the local flora. She was doing it at a time, as she recognised, where a lot of the native bush was being cleared for agricultural expansion, and she could see that it was really important to document it while it was still there. She wrote to Jim that, "You can't imagine how I long to be just independent enough to follow my two desires, botany and writing." But she needed help in doing that, and her long correspondence with Jim Willis served that purpose. She would send specimens in to be identified. He would write back with the names. And the value of those relationships between herbarium staff and enthusiastic naturalists is priceless. So Eileen's collection is incredibly valuable. It includes several species that were the first herbarium records for the state of those species, and several of them are now rare and threatened, and a few are even thought to be extinct. One of the ones that hadn't been collected since Eileen collected it in the early 1950s was the bearded flat sedge, or Cyperus squarosus. But it was actually rediscovered last autumn, April or May, following flooding, the extensive flooding up in the Mallee. And it was only through sending the specimen into the herbarium here and comparing it against the specimens collected by Eileen that the identity of that thought to be extinct plant could be verified. Now, we couldn't possibly talk about contribution to the herbarium without talking about another enthusiastic non-professional botanist and naturalist, Cliff Buglehole. Cliff Buglehole was a farmer in southwest Victoria who took an intense interest in the flora of the state. He began studying plants as a young child, and it said that he could identify and name 60 orchids before he was 10, which is, I don't know many people who could achieve that throughout their entire life. He undertook botanical surveys of the entire state, and it's very unlikely that there's a single track or road in the state that he didn't collect on. He did all of that while making important contributions to the studies of bees, wasps, ants, birds, and fossils as well. In total, he collected about 90,000 plant specimens. 70,000 of them are here, and there are lots of duplicates at other herbaria. And try as they might, it's pretty unlikely that any collector these days will exceed his efforts. 54,000 of those specimens he collected are from Victoria, and they account for 18% of our entire Victorian collection. In a 2002 tribute, Jim Ross, who's another former director of the herbarium, wrote, "Cliff had tremendous drive, energy and commitment, working from dawn to dusk in the field collecting and recording, and half of the night documenting and processing his collections." So it really is a labour of love. This here is Raleigh Black, whose collection was purchased by the herbarium in 1957. So some collections are donated, some are purchased, some are bequeathed. And Raleigh Black was a man of many talents, and he had a huge intellect despite leaving school to start work at the age of 12. He first worked as a printer's assistant, then as a clerical assistant for the Tasmanian Treasury. While there, he undertook memory training courses through the New York Institute of Science, which would have really helped his capacity as a field botanist, where just that visual memory is so important. His interest in the Tasmanian flora began in 1900, and he sought out Tasmanian government botanist Leonard Rodway as a mentor. So over the next 50 or so years, Raleigh Black put together a herbarium of about 15,000 specimens. He exchanged specimens, primarily agricultural ones, with other herbaria and collectors in the US, the UK and Canada. And his collection is so important that Harvard University and Kew Gardens both expressed an interest in purchasing it, but we managed to secure it for our collection here. And I really love this part of this tribute written by Jim Willis, that his brown bespectacled eyes and smiling face beamed good humour, and he was courteous to a fault. One instinctively felt glad to meet him. And for me, I just think that photo of him sort of shows all of that warmth beautifully. This is Ilma Stone. So she was born in Melbourne in 1913. She received a Master of Science from the University of Melbourne when she was just 20. But then she married and didn't pursue botany until later in life. The story goes that she was listening to the radio while doing her family's ironing, and she heard an ad come on from the radio ad from the university in 1959, advertising for chemistry tutors. And she lamented to her husband that they weren't asking for botany tutors, but he encouraged her to just call them anyway. And they welcomed her back immediately, and that was the start of a really long and illustrious career as a bryologist and moss expert. So she described 25 species and had 5 named after her. She published a lot of scientific papers and co-authored The Mosses of Southern Australia with George Scott in 1976. So mosses aren't pressed like the higher plants. They're stored in packets and boxes, and Ilma Stone would use whatever packets she had to hand to store them in. So we have all sorts of lovely little chocolate boxes with a little moss in each little hollow where the chocolates were, and tobacco tins and cheese boxes and things like that. So you get these lovely insights into the lives of the people who collected. Another prolific collector, and this one's a former staff member, Rex Wilson, was specialised in lichens, and that specialisation really diversified and enriched the herbarium collection here. His early career was actually in carpentry, and he was employed as a carpenter on the Australian Antarctic Division in the early 1960s. So that took him down to the Antarctic. And that also marked the start of his lichenology career. He was just fascinated by all the lichens in that landscape. In 1964, he was employed by the Gardens, making him the first lichenologist to be employed by any Australian herbarium. He made several visits to Antarctica and the sub-Antarctic islands, and the herbarium houses unrivalled collections of lichens from those regions, thanks to his work. He was awarded a Churchill Fellowship to study lichens in the Northern Hemisphere in 1970. And so if you do a little analysis, and it's a bit biased by what parts of the collection have been catalogued, but Rex Wilson comes up currently in the top 5 collectors for 4 different continents. He's that prolific. He published nearly 60 books and papers and illustrated many of them himself, and these are some of his illustrations here. So these people, handful of people I've talked to you about now, and the 30,000 other collectors who have contributed to the collection are united by their passion. In describing his own career, Jim Willis wrote that your lifetime's work was your hobby as well. And that was true for Mueller, it was true for Willis, and it really remains true for many of our botanists today. Our ID's botanist, Val Stasiuk, who's one of our most prolific contemporary collectors, I think he's up to about 12,000 specimens, about 80% of those have been collected in his own time, on his after work and weekends, not on paid collecting trips. So the entire herbarium collection, both the Australian and the global components, is an extraordinary scientific resource that underpins the botanical science that we do here, but it's also a testament to human endeavour. The specimens not only document the presence of a plant, algae or fungus at a particular place and time, but they document the determination and curiosity of the people who collected them. So they're botanical time capsules in that very, very human sense as well. Like all the collectors, every specimen in the collection has its own story, and I just want to share the story of this specimen with you. It's a very unassuming little specimen, but it does, like all of the specimens, increase our understanding of global biodiversity and can inform conservation efforts. It also speaks to another part of the collection that really inspires me, and that's this idea of the collection being the work of many hands over many generations. And this tiny little specimen does have a rich history with many hands involved in it. It's Cerastium alpinum, which is known as alpine chickweed or mouse ear. It's a small, spreading perennial plant that grows up in the Arctic and northern Canada and Greenland. And it was collected by Dr. David Walker, who was a young naturalist and medic from Belfast in the late 1850s. But the origins of this specimen arguably don't lie with him. You can trace the origins of them, the motivations behind the collecting of any specimen back through history, probably as far as you like, really. But for this specimen, I'll start with Sir John Franklin. Sir John Franklin was, as you may know, Governor of Tasmania from 1839 to 1843. Before that posting, he had undertaken 2 expeditions to the Arctic to look for the Northwest Passage, and he undertook a third in 1845, but that one never returned. Following his disappearance, John Franklin's incredibly resourceful wife, Lady Jane Franklin, sponsored 7 expeditions to discover the fate of her husband's expedition. The 6th, which was the first to find the missing ships, was a voyage under the captaincy of Sir Francis Leopold McClintock aboard the Fox, which set sail from Aberdeen in 1857. The naturalist aboard the Fox was David Walker, who collected that specimen. He was 19 at the time, so he'd actually studied medicine in Belfast at the age of 14. Following this expedition, Walker and the other members of the expedition crew were feted as heroes. They met the Queen. Walker had his portrait hung in the National Portrait Gallery in London, but then he moved to the US and Canada for the rest of his life and sort of faded into obscurity. So what I love about this tiny little specimen is that it has these rich connections hiding behind it. And you can see how the histories and motivations of all sorts of different people sort of converge in this single botanical document. This specimen is part of their legacy, and its curation and care at the herbarium is part of our legacy. But the story of this specimen doesn't end here. Having only been mounted and databased about 18 months ago, it's only beginning the start of its life as a really useful scientific resource. And it started that journey already thanks to the work of our curation team. So within a fortnight of this specimen being databased and then uploaded to those online repositories I showed you earlier, we had an email from a researcher, Paul Sokoloff, at the Canadian Museum of Nature. Paul is studying the flora of the rapidly warming Arctic, and what he's doing is doing herbarium surveys, so looking at herbarium collections from the Arctic Circle, and then going out and doing field surveys to see what's still there and what's changed. The Arctic Circle, as you might know, is warming at 4 times faster than the rate of the rest of the planet, so it is really urgent work. So it was just fantastic for us that this little, within a fortnight, like I said, of us handling the specimen and cataloguing it that we knew it was being used for really urgent conservation work. So that's what field presses look like when they're out collecting. They often don't get stacked quite that full, but it's they can do. So, as well as all the connections behind the collecting of that specimen itself, I love that it's facilitated these new connections. So we've had email exchanges with Paul. We now photograph specimens from the Arctic Circle and send them to him when we come across them. And so it's yet another sort of collaboration through time and space. So I have described some of the network of people behind the collecting of this specimen and the stories behind it. But when we first looked at it, when it was first catalogued, none of that was known to us. It says Dr. Walker. We didn't know who he was. And it's only through a lot of sleuthing that we could unravel the story of this specimen, a lot of examining handwriting and labels between it and other specimens with the same handwriting, that we could really figure out that that specimen was collected on the voyage of the Fox and then learn about the history of the Fox expedition. But this kind of detective work isn't unusual. It's really what the curation team who look after the collection do every day. To function as a really useful botanical time capsule or a really useful scientific resource, we need to be able to pinpoint a specimen to a particular time and place. And while the contemporary collections collectors are really thorough and give us all of that information, the older specimens, they often don't. And so we do have to do a bit of detective work to figure out the missing information. We need to get really familiar with the collectors, their lives, their journeys, where they went, what they did, and their handwriting to uncover the details. So while we're doing all of this detective work, we can sometimes uncover more than we expect. So one of our curation officers, Helen Barnes, has been researching herbarium specimens collected by John Stuart Mill, who most people would know as a philosopher and not a scientist, but he was both. Unable to pinpoint a mountain given as the collecting locality on one of Mill's labels, she contacted a small museum in Bad Ems, which is a spa town in Germany. The museum not only interpreted the locality information in a way that allowed us to pinpoint a latitude and longitude so we could map that specimen, they also sent Helen an accommodation list. The list, which is from the Four Towers bathhouse, includes not only John Stuart Mill, who has erroneously been entered as Mrs. Stuart Mill, but also Harriet Taylor and her daughter. John Stuart Mill and Harriet Taylor were later married, but this visit to the spa was in June 1839, which is 10 years before her first husband died. So they are known to have had a very close friendship and to have travelled together, but this is the first record of them staying in the same accommodation. And I'm not saying anything untoward happened, but suffice to say that this document, this bit of information, was of great interest to David Stack, who's a Mill scholar at Reading University, who Helen's been corresponding with. So we get these little, possibly juicy tidbits of people's lives, as well as the scientific information. But it's not only in those historical specimens that we get these personal traces of people. And I've been really charmed by a small collection of about 150 specimens collected in the outer western suburbs of Melbourne in the 70s and 80s by Barry Kemp, who's a keen naturalist and botanist and an early member of the Friends of Organ Pipes National Park. His little collection provides a valuable record of the flora of those outer western suburbs before they were cleared for urban expansion. It includes several conservation listed, so rare and threatened taxa that are likely locally extinct now. And so it's a wonderful little record of biodiversity, but I also love the human element of it too, and I'm sort of awed by the care and dedication that people put into their collections, and things like the little carefully ruled pencil lines on this label, just to me, just charming examples of how much care people put into documenting the flora around them. But that doesn't apply universally. So not every collector is so meticulous in their label writing, and we do have an awful lot of labels that look like this. They do evoke so much in themselves, though, these labels, and they give us a lot to interpret, transcribe, translate, and puzzle over for sometimes hours. These labels are all from specimens being databased by a team working on the Global Collection Project, which is an entirely philanthropically donor-funded project working to catalogue that global collection, the non-Australian specimens. But we also have plenty of handwriting challenges closer to home as well. So this label was written by John Dalekey, who was a superintendent of the gardens here, and then later became a paid collector for Mueller. He did a lot of collecting in the rainforests in Queensland, sort of around Rockingham Bay. He's Dalekey's a bit of a favourite for us in the collections team. We sort of have this love-hate relationship with him. His handwritings are notoriously difficult to read. There's no punctuation. His spelling wasn't great. And he throws in bits of unexpected information just to sort of throw you off track when you're trying to figure out what he's writing. So sometimes I think this one might have something about it. He'd often, because the rainforest trees are so tall, he might have to shoot down. He'd use his rifle to shoot down flowers and fruits from the top so he could collect them. And if that didn't work, he might have to resort to cutting down the tree. And he would always express his remorse about having to had cut down the tree. He also, on one label, puts in a little bit of information about his daughter making jam of the fruit from the tree, which are all lovely things, but when you're struggling to figure out what he's saying, it does throw you off the scent a bit. So curating the collection is really a collaboration through time. We handle specimens that are so carefully collected by other people. We read and interpret their handwriting, which can feel really intimate in and of itself. And the people who work with the collection, whether they're the curators, the botanists, the people who mount the specimens, they all leave their traces on the specimens as well. One herbarium curator who has left a lot of traces on specimens in the collection is Doris Sincora. She was a curator and a phycologist, so an algae expert, and a historian of botany. And her skills in interpreting and translating the notes on the algae collections, which many of which are in German, she was German, have greatly enhanced their scientific value. They allow us to interpret them in ways that mean they can be used. This specimen here, which was collected by Wilhelm Tilesius von Tillenow during the first Russian circumnavigation of the globe in 1806, is covered in annotations by Doris. They help us make sense of the specimen, to understand its history and its role as a botanical time capsule. When I come across specimens like this one, I have so much respect and admiration for Doris and the work that she did, and it really is an honour to walk in her footsteps as a curator of this collection. But I'm also awed by the work of Tilesius himself and other collectors like him. So this is an illustration of another seaweed, a sargassum species, that was also collected on that first Russian circumnavigation. This is just a fraction of the illustration and the notes, which cover two sides of about an A3 sheet of paper. In it, Tilesius writes something along the lines of, the day, one can spend all day pulling bunches of fresh seaweed from the water daily, so that the night is barely long enough to write everything down. And so when I see an illustration like this, I picture Tilesius working at night, probably by oil lamp, possibly on a moving ship, doing this exquisite work. And it's really is such a pleasure and an honour to work with specimens like this one. So just to finish up, all of the curation and detective work that we do with this extraordinary collection helps make that picture of past and present biodiversity clearer. And the clearer a picture of past biodiversity, the better we can understand current and future threats to species survival. So thank you. I do just want to finish by acknowledging that so much of the herbarium the work of the herbarium is made possible through the generous support of groups like the Friends of the Royal Botanic Gardens, Melbourne, through our volunteers, philanthropic groups like the John T. Reid Charitable Trusts, and many, many individual donors. So through that support that they give us, the care of this incredible collection is part of their legacy too. Thank you.
If there's little bugs or aphids or whatever?
Good question. So the herbarium extension is what we call where the actual specimens are stored. That's what we call the clean side of the building. We also have the dirty side of the building, which you're in now. And the barrier between those is a biosecurity freezer. It's a big walk-in freezer that has the door on either side. And specimens and all paper materials going into the collection spend a week in there at minus 23 degrees, and that kills any things like aphids, any museum pests like soakers, silverfish, carpet beetles, cigarette beetles that like to eat the collection. So that integrated pest management is a very important part of the work that we do in looking after the collection. Thank you. All right, I think we'll finish up there. Thanks so much for coming.